Welcome back to the Pager podcast. So SARS-CoV-2 sequencing has provided us with lots of information regarding the evolution of the virus and, for example, how it spread. This episode, I'm joined by Professor Sharon Peacock to talk all about the sequencing efforts in the UK. Professor Peacock is a Professor of Public Health and Microbiology at the University of Cambridge, where she runs her own research group. She also holds a leadership role in pathogen genomics at Public Health England and more recently became the director of the COVID-19 Genomics UK consortium, COG-UK. COG-UK was founded in April 2020 as a collaboration between UK public health agencies, a number of NHS agencies and universities, as well as the Wellcome Sanger Institute. And so far it has sequenced over 1 million viral genomes in the UK. Our conversation mainly focuses on the logistics of sequencing the virus in the UK and the utility of doing this on such a wide scale, as well as the data that can be gained on a more local level. We talk about monitoring and defining variants of concern, as well as how pathogen genome data links in with epidemiological data and the data from human genomes, for example, and the extent to which we can really predict from genetic data what impact a virus might actually have in the real world. Towards the end, we also turn our attention towards the more slow-burning threat of antimicrobial resistance, so stay tuned for that. Professor Peacock, thank you so much for joining me today. It's such a pleasure to be chatting with you.
George, it's an absolute pleasure to talk with you, so thanks for the invitation.
So I'd like to start off by talking about the COVID-19 Genomics consortium in the UK, COG-UK. How did this get set up? Because I think it's quite an interesting story.
It is interesting. It started really back in sort of late February, early March 2020. And if you cast your mind back to then, there are actually quite a small number of diagnosed cases of COVID-19 in the country. We're kind of at the very, very start of the pandemic. But we've been strong in the UK in pathogen genomics for, a decade or more. And it's no great surprise to anybody that if you have a new pathogen emerging, then it's going to undergo evolution, natural selection for a virus that would develop characteristics that could give it an advantage over the kind of original virus. So a group of us knew that sort of sequencing the virus over time, if we were going to be living with this virus for any length of time, is going to be important to predict its biology. And towards the end of February, beginning of March, I was talking with Sir Patrick Vallance, our government chief science advisor, to say, this is important. And so really what happened next was I sent a few emails out to some colleagues who work in pathogen genomics to say, I think we're going to need to set up a national sequencing capability. Do you agree? And everyone was very supportive. And at that point, by mid-March, we called a meeting of about 20 people, 20 experts. We were given a room at the Wellcome building in Euston Road in London. And we basically sat down over the whole course of the day. We worked out a blueprint for how we would develop COG-UK. Now, scientists will often usually get down into the minutiae of their science. But actually what we did, we structured it around how would you do this, who would do it, where would we do it? So by the end of that day, we had a blueprint for what COG-UK would look like. And there was a lot of debate about where we would do sequencing, but we kind of decided that the best model was bringing everybody in who had the capability, involving public health agencies and academia and the Wellcome Sanger Institute. So we wrote that up and we put it on Sir Patrick's desk, and within a very short space of time, we were funded to get going. So by the 1st of April, we were an entity. So we got started by really pulling in the coalition of the willing.
And am I right in saying that actually this was a gamble in a few respects early on, because there were some out there who said, okay, is there any point in sequencing a virus if we don't know it's going to change particularly? And there were also people saying, okay, this isn't, particularly in mid-February time, this isn't something that's going to be significant in the UK at all.
It was a bit of a gamble, I guess. My sense was actually it's better to get organised and get ready for anything that's going to happen. I mean, the worst that could have happened, I suppose, is that we could have sequenced a lot of very similar viruses and ended up with not very interesting scientific papers. But in the best situation, which is what we found ourselves in, actually, we'd be one of the best prepared countries in the world for sequencing and provide a huge amount of information to the world on viral genomes. You're quite right, George. There were some critics, and the criticism was around the fact that, the virus at the time when we started sequencing was actually really quite similar to each other because it only just emerged, and so all of the kind of the progeny was highly related. But starting that early was important because we had to get the systems and mechanisms up and running. So we needed to get that structure, we needed to get kind of governance structures, we needed to get ethics and the legal framework and data sharing agreements. All of that was required. It was then a question of, what we could learn. And, even as early as the very first wave of the pandemic, we did learn some really, really useful bits of information. So, for example, a study looking at the viral introduction into the UK. At the time, during beginning of wave one, we were worried about China, we were worried about northern Italy. What the genomics told us was actually much of the virus introduced into the country was from Spain, France, and Italy. So we were kind of that was a really important early lesson, particularly around travel. So yeah, there was some critics. By the time we got to the point where variants of concern emerged, and there was kind of like a sort of 4 variants of concern emerged relatively close to each other, there were no critics. But if you are a scientist or anyone really, we all face criticism, and we just have to make our own decisions based on what we think is the right decision, and then stick with it. Might lead to some sleepless nights, but that goes without saying that if we think something's important, then that's what you go with.
Exactly. If we only did things that were guaranteed to succeed, then we'd be guaranteed to be not doing a lot of things that could have had widespread beneficial effects, as COG-UK has.
I think that's right, George. I think if you don't take risks, with some of the decisions you make, you can stay safe. But in retrospect, we're all really pleased as a consortium that we got started when we did.
So you've mentioned one aspect of this already, which is tracking the origins of viruses or different variants introduced into the UK. Could you talk a bit more about actually why genome sequencing is so important in the pandemic?
Well, it is all to do with understanding the implications of the virus evolution. So we know that this particular virus, the SARS-CoV-2 virus, it mutates once or twice a month, during replication in humans. And that's basically, it's got 30,000 bases in its genome, and it makes a mistake. So another base is put in its place. And some of those, a small number, not many. I mean, most are not. They don't make the virus fitter or deleterious to the virus. But some of those will make the virus potentially fitter, being able to transmit more or, escape our immune system. They change the function of the virus. So what we really need to do is track that evolution. And what we're asking, I think the key question that we're asking here is, do we understand the mutations that arise that are associated with evasion of either our immune system, which means that the virus can infect us twice or more, and escape the effects of our vaccination? That's absolutely top priority. So we need to look at vaccine escape and reinfection. The same is true for therapeutics. So, for example, the virus could develop resistance to antiviral drugs and also monoclonal antibodies. And so we have to understand what mutations mean when they arise and whether they could lead to vaccine escape. Just looking at the mutation doesn't tell us that information. We have to go and do the biology. So that's my top application, really. And looking ahead, we need to keep sequencing the virus for as long as it's causing us trouble, because we need to potentially tweak our vaccines. The vaccines are very effective at the moment, but in the longer term, we may need to change the vaccines as we do for flu vaccines over time to keep them effective, and switch our therapeutics around. So that's very important. Now, actually classifying a virus into a virus that's not of particular concern, or one of the 4 variants of concern, also allows us to really understand the biology. So it's like grouping things. If you can group a virus into, say, the alpha variant or the delta variant, it gives you a framework for saying, is this variant more transmissible? Is it escaping immunity? Is it causing more severe disease, or perhaps even sort of less severe disease? And is it sort of causing problems with our diagnostic testing? And so that categorization is the kind of like the very beginning of that kind of chain of investigation, if you like. We also use sequencing to really understand how the virus is transmitting. So studies have been done in hospital, for example, or prisons, which can actually identify where lapses of infection prevention and control have occurred and can help that to improve. But it's also been quite instrumental in understanding introductions into the country. We've already mentioned that briefly, but when we were sequencing when Delta first emerged, then knowing that had been introduced several hundred times into the country, I think was an important piece of information. So if we are sequencing quickly enough to know what's happening elsewhere, for example, even where places are not doing sequencing, we can understand what's being introduced and what's emerging elsewhere. So there are really multiple reasons why we need to keep doing this, but the bottom line is that it does lead to policy changes, and it does lead to public health action. So it's like it produces actionable information. This isn't just like stamp collecting or counting how many variants do we have. It's actually guiding the actions that we take.
Definitely, and I think that's the best way to look at it. You've mentioned quite a lot there about its utility, and I'd like to just perhaps focus in on them one by one for a little bit of time, and perhaps first talk about the dynamics of transmission. And I wonder, was there anything that the data coming out actually showed us that was unexpected relating to transmission, or at times was it just reinforcing what we perhaps already suspected via other models?
Well, we certainly knew that the virus was a respiratory virus. It's going to be transmitted from person to person. I think that there were some surprises, which I think improved practice. I think a good example of that is work done in Cambridge, where in the hospital, where some outbreaks were being investigated, and this one particular outbreak was in people who were having renal dialysis treatment for kidney failure. And there was an apparent outbreak in these particular individuals. But actually, when you added the epidemiological information to the virus information, and that is always an essential partnership, actually, you can't just gaze at the genomes. What was discovered actually was that it was the hospital transport where people had been in contact and had transmission events, and it wasn't the renal unit. So those sorts of insights, as just one example, but those sorts of insights can help you understand how you need to improve your care of patients. And so as a result of that, changes were made to the way people were transported into hospital. So that's just one example of just in one place what colleagues learned about transmission.
And I think that's a really good example, because when I look at reporting on transmission, it seems like there are lots of very plausible explanations for where transmission can occur, and actually being able to narrow those down and not have to necessarily intervene in all circumstances, because obviously there are certain kind of levels of contact that are essential. People going into hospital, having medical treatment is essential contact, we would call it, but actually there are other areas where that contact might not be as essential, or certain barriers can be put up to that contact. And if we're focusing in on the wrong area, then actually we're at risk of causing almost damage by limiting contact inappropriately in some areas, whilst letting it carry on perhaps on hospital transport.
Yes, and genomics is simply one tool. Actually, it's not the ultimate answer, but it does give us insights into areas where we can improve infection prevention, control, and patient care.
And I think this takes us on to talking a bit about actually how the data from genome sequencing links in with other data. And I'd like to first touch upon looking at the impact of the virus on, I guess, people's health. When we're talking about, for example, the efficacy of therapeutics, or even, for example, the fatality rate with the virus, what sort of information are we having to gather with genomics, and what are the logistical hurdles to doing that? Because this is very different data.
Well, I've mentioned before the epidemiological data, by which I mean, who is the sample from, what was the date of the sample, where were they, and their particular information. Did they go to hospital or not? Were they unwell or not? Were they ventilated, etc. Is really important. Just taking your question about disease severity. And so evolutionary biologists have proposed that there's not much advantage to the virus to become, cause more severe disease. When you think about its perpetuation, it needs to be transmitted from person to person in order to perpetuate or avoid your immune system. And there's quite a lot of debate about whether the virus become, less virulent over time and become, sort of basically like the common cold. But actually, for two of the variants, Alpha and Delta, studies have demonstrated that they are more likely to cause people to go into hospital. And for those studies, what you're really doing is you're looking at people with different variants, and so that involves sequencing. And so you may, for example, with the Alpha variant, compare people infected with the Alpha variant compared with people who are infected with non-Alpha variant, and actually see sort of adjusting for their comorbidities for serious disease and their age and so on, whether people infected with Alpha or non-Alpha are more likely to be going into hospital. So really what you're doing is then sort of studying a sort of cohort of people in a certain time frame and a space to say, were you more likely to go into hospital if you get Alpha variant rather than not, having adjusted for confounders for serious disease. And then, and so that has been done for several variants. So are people more likely to require oxygen and have some form of ventilation, and do they unfortunately die as a result? So those sort of studies, it's really a combination of detailed clinical information joined up with the genomic information, which allows you to get to the ultimate answer. And of course, it has to be suitably powered in order to make a statistical sort of answer, really, a powered answer.
And I wonder, as a passing thought, whether actually the debate over whether the virus, it's advantageous for the virus to become or to cause more severe infection or milder infection actually is really kind of a false argument, in that it depends on the context. I can imagine a scenario, for example, in which actually in a state where there's not much social contact, in lockdown, increased severity would mean increased contact with other patients and healthcare staff, and could increase spread. Whereas actually in a more of an open time, then actually there might be more spread from somebody with milder disease who is allowed to go outside and make more contact. So the context seems to be quite important here.
And I think if the causing more severe disease is linked to increased transmissibility, you can see how they could be co-linked, really, those biological characteristics. But I think it's really difficult to predict actually which way the virus will go. And some people have suggested that the virus has gone as far as it can go and it won't be evolving any further. Others have said it's just going to become like the common cold. I actually bring to the kind of discussion a sense of humility, actually, because we don't know. We're learning every day. We learn every day that we manage this pandemic, and we have to be willing to change our mind. But when people say, what's the virus going to do next? Is it going to become like the common cold? My sense is, we need to learn on the job. We need to be flexible in our thinking about this. And I don't think anyone has got that level of crystal ball knowledge. So, obviously, as a scientist, I read quite extensively what other people think. And I don't think anyone has got, a categorical answer as to what's going to happen next. And that's why we just need to keep sequencing, actually.
It seems to me as if it's... people may be right, but they may be right for the wrong reasons. If you're guessing and you turn out to be correct, then that's not necessarily justification to trust you the next time around. So you need more than just correct answers in the past in order to have, I guess, trust in what you're saying.
Well, we place understanding of the past on viruses that are either not related or closely related to SARS-CoV-2, but they're not SARS-CoV-2. And so one can have a reasonable, prediction. You can make a prediction of what's going to happen. But I think the proof is actually in what happens, over time. And as we approach the winter, even predicting what might happen during the winter, actually, in terms of, the co-circulation of flu and so on, it is modelling at the end of the day, which we often use actually for trying to think what might happen next. Is at the end of the day modelling, and we have to wait for the truth to come out by living the next day and the next day and so on.
As the famous saying goes, all models are wrong, some models are useful.
I'm sure that will displease some of your listeners, but there you go, it's out now.
Okay, and I guess the next step or a step that could be done at the same time with looking at this epidemiological data and genomics data is looking at information that is not necessarily routinely collected as part of a patient's care, but could be very useful for looking at the kind of spread of the virus and its effects on, for example, the immune system. Has there been very many efforts to combine genome sequencing done by COG with, for example, antibody titers, patients, I guess, particular kind of immune predispositions, or even their own genetic sequences?
That's a really good question. So, we do one level of analysis, which is what is the variant and are there outbreaks and so on. And that's kind of like a public health response, and it gives actual information. But what you're talking about is critical. And all of the information that we generate is actually freely available. So we provide it both in kind of readily digestible forms in websites and so on, but also colleagues can go into our database and actually acquire the information. So working very closely with virologists, there's a consortium called the Genotype to Phenotype Consortium. We work with them very closely to say, this is the genetics of the virus. What does the virology look like? And we also work very closely with the UK COVID Immunology Consortium. So that data is all available. And in fact, all of our genomes are put into public databases. So that is really important. And we are just one small part of a bigger science ecosystem. We also work very closely with the Office for National Statistics work, with REACT, and all of the national core studies we aim to support. But you mentioned human genomes, and that's is going to be particularly interesting. So I'm not aware of any studies where somebody's taken 1 million. We've got 1 million viral genomes now, more than that in the UK, with human genomes, and tried to understand how the two might interact. So if you had a combination of viral genomes, human genomes, and very detailed clinical information, could you work out, for example, if there's an interaction between some people and some particular viruses? Could you use the information that you've got to actually predict, not only from the host genetic susceptibility, but actually from the viral genome, what will happen to a person once they become infected? Effectively, you then start to think about stratification of care, so that you treat the person with kind of the right drug at the right time. So that work is just ongoing. Actually, we actually, from the very first meeting, going back to the Wellcome Trust back on the 15th or so of March in 2020, we had representatives from Genomics England there. Mark Caulfield was present. And so we made a commitment really early on to link viral genomes with human genomes, to do that kind of very detailed evaluation. And a real discovery science, actually cutting edge discovery science. We're now working with Genomics England and with the SARIC team, and also Health Data Research UK, HDR UK, so that we're bringing all of that data set together. So we're putting this into what's called trusted research environments, which is where you can have data securely stored. And then people can apply for access to those data sets. So we are bringing together human and viral genomes that people can actually apply to access to analyze. And we're hoping to bring together more information, such as information on serology, immunology, and so on. That's the next level of discovery science that will still be going on, hopefully once the pandemic has largely calmed down and left us. But that's really exciting, actually, and that is cutting edge. So developing the techniques and thinking about how machine learning could actually use information in real time, potentially, to predict patient outcome, for example.
My suspicion, and correct me if I'm wrong here, would be that actually some of the privacy concerns, which I think you've gently touched upon, relating to actually having to apply for access to these databases, those are perhaps one of the significant barriers to doing this, because after all, the samples that we take of COVID-19 contain human DNA and human cells in them in the first place. It's almost as easy a kind of access to samples as you could get within the medical sphere.
Yeah, I have to say that the viral genomes that we release don't have any human data in them, so you can't actually predict where the virus comes from. But you raise a really important point, because people who are providing their human genomes or their viral genomes, they need to know that that's being looked after securely. In the UK, there are very advanced sort of thinking and systems in place to protect patient confidentiality. And with the virus in our database, which is referred to as CLIME, which is a cloud infrastructure for microbial bioinformatics, these are all anonymized. So it has a sort of a number associated with it, but not a name. And we cannot link patients' NHS numbers, for example, with that. That is all held by the appropriate authorities, for example, the public health agencies and the NHS. But you're right, we have to think about privacy. And we are fortunate to have organisations such as Health Data Research UK, actually, who pay a lot of attention both to privacy, but also patient and public participation, because people will want to know what's happening to their genomes, and there are public and patient participation panels in, for example, Genomics England and COG-UK, which is absolutely right. But it provides people with information about what's happening to their data and how it's being used. But also telling people how the data is being used. I think being completely transparent about that is important in gaining people's trust.
Definitely. Are individual data points given, I guess, an identifier that allows you to track whether the same person has been infected multiple times? Because that's quite an interesting question going forwards when we're talking about, okay, are different variants perhaps having some sort of immune escape? Or even actually linking into what we were talking about before, is it more related to the, for example, the genomics or the immunology of that individual person that is leading to that second infection?
Well, certainly public health agencies will be able to work out if somebody's had more than one test. So it would be possible for them to basically deduplicate the information and say who's had more than one test. So that's definitely possible, and that's important. Thinking about the way that the virus evolves during kind of longer term infection, I think that's a slightly different question, and that'd be in the context of a research study with sort of the appropriate ethical approval, where a virus who's, for example, infecting somebody who can't clear that infection, perhaps they have a problem with their immune system. If they have chronic infection with COVID-19, then somebody would have relevant ethical approval and patient consent to take that virus repeatedly over time. So if you want to look at sort of really evolution within a human host over time, then taking that virus repeatedly is kind of the best way of looking at that. And people such as Ravi Gupta in Cambridge have done that work and shown how really the sort of longer term infection, it's like a training ground for the virus. The mutations will emerge and over time. So I think that there are two different questions, but you were talking about immune evasion, and the other thing to really do is to think about it on a population level. So if at any given time point you tend to target your sequencing to people who've actually had the vaccine but then actually get infection, or have had an infection then have a second infection, targeting some of your sequencing numbers, the kind of number that you can do to that population, is like enriching for people that may be the people who get infection by a variant of concern, which is avoiding the immune system. So targeting to vaccine failure and reinfection is an important one important use of our sequencing capabilities in the country. And
I wonder, just moving on, how we go about labelling what is a variant of concern. In my mind, I have this spectrum. I've written it down on a piece of paper where we have the genetics of the virus on one side, and then we have the phenotype of the virus on the other side, and its behaviour in people. And on one hand, we could look at the genetics and say, does this look like it could be concerning? On the other hand, we could wait for it to have its clinical outcomes, which are proved to be concerning. How do we think about this?
Well, the way that this is done, and I agree that it's the weight of evidence, isn't it? But the way that it's done at the moment really is that there tends to be kind of a watch list of variants that could be potentially important, and that list could be quite long. And that could be a virus that's spreading around the world quite quickly, or appears to be associated with surges in certain countries, or in regions. And maybe from soft intelligence or from reports from the newspaper. So if something is of concern somewhere, then that could go on the watch list. But it makes it onto kind of a variant under investigation or a variant of interest. Those terms are kind of used interchangeably really in different countries. When it looks as if there is a signal, for example, that something appears to be more transmissible. But you don't have enough evidence at that point to know whether it's a variant of concern, and it may well have mutational signature, which makes you've observed in other variants that are problematic. So a variant under investigation really kind of marks it out for saying we're going to need to watch this really quickly, really carefully, and see whether it does appear to be more transmissible in populations, whether it does appear to be causing more severe disease or not, and also looking for vaccine failure. So it then goes under intense study whilst it's a variant under investigation. So when the weight of evidence is such that it's shown to be more transmissible and or immune evasive, it then becomes a variant of concern. So it moves up. Now, in the US, they have a higher category of that. That's kind of a pathogen of a variant of high consequence. And that is when a variant is considered to be, fully resistant to vaccines. We haven't seen anything like that at the moment. So coming back to the variants of concern, we have 4 variants of concern at the moment in the world, and we haven't seen a new one just recently. But the level of evidence, it can be really quite challenging for public health authorities to take the step from a variant under investigation to a variant of concern, because there's actually quite a lot of implications for calling something a variant of concern. But what's the level of evidence at which you say it's actually shifted over? And I think if you are too conservative to call everything a variant of concern, then that would be quite challenging, trying to monitor those with increased screening, with surge testing and so on. So we're learning more as we go along, but the evidence can actually take weeks or months. And of course, you've mentioned the biology. You have to look at whether something actually escapes the immunity through neutralization assays, which can take actually weeks or months to complete. Perhaps the challenge for us then really is to say that we need to be able to get to those answers quicker than we do at the moment, because it does take us quite a while to be sure that something is a variant of concern. If we had quicker ways to get that information, then that would be very helpful.
So what you're saying is really that the concerning aspect is born out of epidemiological data rather than necessarily just looking at a sequence early on. Could this be possible because this would be perhaps the earliest COG-UK has, I think your aim is to sample 10% to sequence 10% of infections per week in the UK. A signal from a variant could appear very quickly, and much more quickly than this data later down the line pointing that it's concerned. Do we have this, I guess, ability to look mechanistically at the genome?
Well, the epi data is important. So if you see a surge of cases, but you've actually got to be sure that they haven't all been to the same party, or that isn't, because we do see sort of outbreaks where actually it's the social behaviour that's really key. So epidemiology is really helpful. Looking at the genome gives us important clues. So a lot of experimental work has been done to look at sort of mutations in kind of each point of, say, the spike protein, to see what effect that will have in neutralization. But you still need to do the virology and do the neutralization assays to be sure, because different combinations of mutations can lead to a different phenotype. And even now, we don't know what a lot of the mutations actually do in terms of the biology outside of the spike protein. So there are, there's quite a lot of information to allow us to feel concerned about a particular mutation, but even then you don't know whether this is going to turn into a variant of concern actually. So it's that combination of mutations that it's difficult to predict what the kind of the end result will be in terms of how the virus behaves. So I'm not sure that's a completely satisfactory answer. It'd be great if we had something that we could look at the genome and add a bit of extra information in, and there you have it, you have the answer. But it remains more complicated than that, and it's a combination of every piece of evidence put together.
I spent quite a lot of time talking in the podcast about how mechanisms are only point you in the direction that you should be looking in. They certainly don't give you any hard answers. And I think your point actually about that acting has consequences is very salient as well. We can forget that actually the interventions that we have are very consequential on, I guess, public behaviour. And the boy who cried, well, it's a perhaps a poor example, but the boy who cried wolf comes to mind. If you keep clamping down on variants that aren't concerning, then there's a risk that actually people will respond in a less serious way when actually one does come along that is concerning.
I would say a couple of things about that. First of all, all variants of concern can be controlled with the usual, washing your hands, covering your face, keeping your distance, and fresh air, ventilation. And so in many ways, we shouldn't get too distracted by variants. If you want to reduce a disease, then they still absolutely apply in the same way. But I think one of the things that has concerned me somewhat is that not so much recently, but particularly around the period when we were seeing variants of concern, there were lots of really scary media reports about this variant or that variant being found in different countries without the full facts about whether it's something to be frightened about. And that can be really quite alarmist to people. And I think there's been a tendency in some quarters to actually kind of hype the concern and worry about variants. When variants are predictable and expected, you can still, do the kind of preventive measures to reduce the risk of infection. But I think some of the alarmist reportings around the variants, actually many of which haven't turned out to be particularly troubling, is unhelpful. We need to be more measured about that, so that people don't get alarmed, but also don't just switch off, because if people think, it's just another variant, I don't have to, yet another one. So I think balanced reporting is really important, and we do have a very good system in the UK with the Science Media Centre, who actually are a very good interface between scientists and the media, trying to get really accurate information out there into the media, papers, etc.
And certainly nuances and balance is something that we should be striving for in reporting. In our last few minutes, I'd like to pivot slightly, actually, outside the world of COVID, but actually to a related topic that you've been an intricate part of for many years now, which is looking at antimicrobial resistance. And perhaps the lessons that we've learned in COVID and sequencing could apply in this area. This almost suffers from the opposite issue to what we've talked about before, in that in some areas there is not enough seriousness about the consequences of it?
There's a lot of work going on at the moment in quite broad terms, looking at what we've learned from COVID-19 science that can be conveyed to other diseases, not just AMR, but also AMR. There's the Academy of Medical Sciences and the Department of Health are working together, for example, to develop a workshop in exactly this. What have we learned from COVID-19 that could actually help with AMR control? And that discussion will range from clinical trials, vaccines, diagnostics, and sequencing. I mean, in terms of genomics and AMR, there's quite a lot that we could really consider. I think one of the perhaps unsung heroes, as it were, of COVID-19 is really sewage analysis for SARS-CoV-2. So actually, there's sewage analysis right the way across the country on a regular basis at the moment, which shows the gene copy number per volume of sewage. And it can help you to understand where the disease is surging, even if you're not doing any testing, like clinical testing, PCR testing, lateral flow, etc. So that also captures undiagnosed people who are asymptomatic, who haven't had a test. You can also do sequencing of that sample. So detection is a molecular test, but you can do sequencing to look for variants of concern, variants under investigation. And if you were to look at this as one example of how you could learn from COVID and take this into AMR, it would be an extraordinary opportunity to start to look at AMR more holistically. So at the moment, when we think about antibiotic resistance, we tend to base it on samples that have come from patients in hospital or have been tested for an infectious disease. But we can like SARS-CoV-2, we can carry bacteria that are resistant to antibiotics quite harmlessly as well. So in order to understand, for example, the spread and the burden of antibiotic resistance in our community, which we don't really have a good handle on at the moment, we could use sewage. So every sewage outlet from hospitals could be tested for example, resistance to carbapenem drugs, which is one of the kind of drugs of last resort. People, care homes, populations, you could actually start to get the very first, very accurate and detailed assessment of the extent of carbapenem resistance in the community. And based on that, you could start to look at interventions and controls. You could start to look at sort of where you'd be within prescribing, for example. So I think that's one area, one of many actually, where we can take our learning from COVID-19 and then put that into AMR. And the meeting at the Academy of Medical Sciences and Department of Health, that's going to be really exciting because that's going to, as I say, really explore the whole area of surveillance, diagnostics and so on, and what we can learn to really improve on AMR. But we have to kind of take this incredible science innovations and apply them to other problems. I guess AMR is what you might call a sort of smouldering pandemic, and there's been a lot of antibiotic use in hospitals with people who are seriously ill with COVID. Of course, if somebody's seriously ill, you're going to treat them with everything you can to try and save their lives. But AMR is around. We're just not talking about it very much at the moment because we have other things to talk about and think about.
We talk a lot about kind of cost effectiveness within medicine, and I was curious as to how we go about thinking of this in terms of I guess prevention of events, and for example, a prevention of an outbreak of antibiotic resistant bacteria, because this doesn't seem to be as straightforward as the other, I guess, cost effectiveness studies we might do, where we can see, okay, what's the difference between one patient group and another, because we have this uncertainty of whether actually something would have occurred in the first place and quite how large an outbreak could have been.
Well, I would say that there is some data there. So certainly for methicillin-resistant Staph aureus, or MRSA, there's been cost-effectiveness modelling done to show that if you, for example, do routine sequencing of all MRSA and detect outbreaks early, that actually the cost of sequencing, which is relatively low now, actually it's cost-effective to do that to prevent extension of a larger outbreak. And of course, then there's the issue of just doing a better job. So there may be times when actually, spending the extra money is the right thing to do, actually, because it actually treats patients, better.
Definitely.
Another area where sequencing is clearly effective is, for example, sequencing of Mycobacterium tuberculosis, the cause of TB. So actually sequencing to predict resistance based on the genome is clearly quicker than sort of phenotypic testing, particularly when there's quite complex resistance in that organism, but actually more cost effective. So cost effectiveness is an important part of thinking about how to use technologies. But I think the evidence is growing for sequencing. And I think the third example I would give you is foodborne outbreaks. So the food distribution patterns that we have across the world now are immensely complicated. And actually, sequencing will actually make a connection between different countries or different places in the country in a way actually that phenotypic testing couldn't even do. So there might be circumstances where you just say, well, this gives us so much more visibility of a problem. A typical, an example here, in 2019, there was a listeriosis outbreak in hospitals. It was reported in the news. I'm not saying anything that's confidential. And so there were Public Health England routinely sequence Listeria monocytogenes. Now, Listeria is a sort of a bacterium that can be in food. Mostly, it won't cause any problems if you're healthy, fit and healthy, or it might cause mild gastroenteritis. If you're immunocompromised or pregnant, it can cause quite severe disease such as meningitis, septicemia, and about 20 to 30% of people die. In this particular outbreak, there were two isolates that Public Health England found were very highly related on sequencing. They were from different parts of the country, and then a third came along that was also related from another different part of the country. And actually, it was from sandwiches that were distributed to hospitals that were contaminated in a sandwich factory. That led to a massive investigation. The plant was closed, and Listeria from that factory matched the patients. Now, in the end, there were a handful of cases, and given that we have around 160 cases of listeriosis overall in the country, it's like you could never ever spot that an outbreak was happening, and that was nipped in the bud. So even if you paid, if the cost of that test was actually quite high, it actually saved huge amounts of morbidity, actually, and potentially fatality by nipping that in the bud early. So, there is an importance of cost effectiveness, naturally, but I think that story about listeriosis really indicates to me that it can, it can save lives, actually, it put quite simply.
I think one thing it highlights to me is, I guess, a core thing of looking at cost effectiveness is making sure that you have actually accounted for all of the costs and all of the benefits. And again, if you only have a snapshot of one area, then actually, as you said, with phenotypic testing, you might actually miss what has been prevented or what could have happened or what is perhaps linked in a completely different area, in a way that with non-transmissible diseases, that isn't really the case. And I love your example with TB as well, because I think that's another great. TB is notoriously difficult to grow or slow to grow, and actually sequencing, having something that can give you results much earlier on and inform, I guess, patients' treatments for something that is still a very concerning disease and transmissible as well, is very important.
Yeah, absolutely, George. The thing with TB is that there are molecular tests to look at some of the first line treatment, so you can get that answer really quickly. But once you get into second line treatment, some of the drugs that are used for multi-drug resistant TB and extensively drug resistant TB, it does take weeks or months to actually get close to whether the organism is susceptible, and to be able to actually predict that is something that is very powerful, because the sooner you treat somebody with the right therapy, the sooner you can start to get control of their disease and the potential for spread to others. But the nice thing about that is that you can also got the genome data to look for outbreaks and relatedness of TB isolates to each other. So it kind of gives that added benefit. And I mean, the UK routinely sequences all MTB now, because the case has just been shown that it's effective and worthwhile.
I think at this point, I'm aware that we're out of time. The time has flown by, so I might have to draw things to a close at this point. But it's been a really lovely conversation, and it's hopefully given everyone, and it's certainly given me a lot of food for thought. I guess going forwards, this could play quite a significant role in healthcare. So thank you so much for joining me.
Well, George, it's an absolute pleasure, and thank you for doing these podcasts, because you're doing an important job to explore subjects that I think your listeners are really going to benefit from. And just finally, a big thank you to my consortium. More than 600 people, mostly volunteers. So thank you for all your contributions. So thanks all round.
Thanks for listening to the Pager podcast. If you've enjoyed this episode, then do share it with a friend and come back to listen to our other episodes. As ever, we'd love to hear your feedback, and you can reach us on social media at pager_podcast, and you can also email us at pagerpodcast@gmail.com. We hope to see you again.